Ah, that'll be fun.
That's what it is.
All right, let's go ahead and get started. I will put up a updated schedule up on the Blackboard. I'll try to get to it by tomorrow. But just so you're aware, we move into HIV on Friday. So we'll go through HIV Friday, Monday, and Wednesday next week. Obviously, Friday next week is posters. If you want to come early, I will have coffee available ahead of time to fuel you all for that period of time. So I will make coffee. Would anybody? I'll bring some fruit. Okay. Greenbush.
Yeah.
I will run my ass over to Greenbush early in the morning and go pick some up. Okay, so if you want to stop by a few minutes early beforehand, I'll just have it all sitting in my office. Come on by. Oh yeah, the actual poster session is downstairs in the atrium, so just on the first floor. So, yep. Okay, so that's one. I've seen about 2/3 of the posters, so if you've got a poster, try to get it to me by this evening so that I can look over it and get it back to you by tomorrow. I think that's it for right now. I think that's it. And then I should have exams graded. If everything goes well, I'll have the last question graded tonight, and you'll see grades starting to come up either tonight or tomorrow morning. Okay? All right, so I think that's about it. Are there any questions? Okay, so thank you to Shania, Taylor, and Angela for asking a question yesterday on the paper. Did everybody get the email I sent? So, I'm surprised. I usually don't get emails back from people at big universities that quickly, but it was really nice. So, and I think that actually made some clarification, but I still disagree with how they report it, but that's a point of contention. Don't ever tell that to the person who's written the paper. So what we're going to do is we're going to talk today about this paper on determining mutation frequency of Ebola virus, and then also talking about how to exploit that in therapy. The way that I want to do this today is actually I am not going to do the majority of the talking. Yay! So,
Your voice is so soothing.
no, it's not. It's not. It sounds really, really horrendous. So, kind of like a cat scratching. All right. So here's what I'm going to do. I'm going to give a quick couple of pieces of background on I don't know if it's ribavirin or ribavirin. I'll honestly admit I don't know on this one. We could ask Google, I suppose, if we wanted to. But I'll give a little bit of background on this drug, and then what I'm going to do is I'm going to break everybody up into groups of 2, and each group is going to get a figure or a part of a figure or something to explain. Yay! All right. So, ribavirin, nucleoside analog. In other words, it looks like an A, a G, or a T, or a C. Particularly, this one is a guanosine nucleoside analog, so it looks like G. We will see these types of drugs come up again when we talk about HIV. Actually, probably more than any other class of drugs for HIV, the nucleoside reverse transcriptase inhibitors are the most common. They look like nucleotide bases, and their incorporation into the genome oftentimes leads to a mutation or a stalling in the replication process. Okay, so we'll come back to this. But on the right here is a picture of guanosine, and then on the left is a picture of ribavirin, and you can see that they've both got the ribose base. If they were modified, they would have triphosphates on here as well. The difference is just this base structure here. This one does not get recognized properly when you're trying to make a copy of it on the other side, or make a complement of it, and that's what leads to the mutations. Okay, so the potential benefits of using this then are that it can improve it can cause mutation of the virus, which then leads to what they describe as error catastrophe. What could somebody describe what they mean by error catastrophe? Jonah, go ahead.
Mutations in the genome that make it so that the virus can replicate.
Yeah, or take it to a point where there are so many mutations that have occurred that it can't overcome, at least in the protein sequence, the mutations that have occurred. Exactly. So increase the rate. And this is just an example of how ribavirin is used to treat hepatitis C infections. We're not going to focus on all of this, but what I really want to do is just kind of focus on what's going on right in the middle here. And so there are a few ways in which ribavirin is thought to work. One of the ways is just stalling or inhibiting the actual replication process itself. As you know, many DNA polymerases have proofreading ability. RNA polymerases don't, so I'm not exactly sure how this mechanism works now that I'm thinking about it, but there is something that can stall the replication process. The way that will be discussed in this paper more is this methodology, mutagenesis. So increasing the mutation rate of, in this case, Ebola virus as a way to produce viral particles that are no longer infectious, that no longer would lead to a productive life cycle. Okay. All right. That's all for me, Jonah.
So I tried to look this up, but I couldn't find anything. Is there any way that the virus will interfere with the host cell's transcription? Just potentially mess up some proteins a little bit?
Quite possibly, yeah. Yeah. What you might expect, though, actually, I'm gonna be cautious with this. Is it a possibility? Yes. Have they looked into that? I'm sure that they have. If it's an approved drug, I'm sure that has been looked at. Now, whether ribavirin is selective for RNA-dependent RNA polymerases, that would be the question that we'd really want to ask, or if it's for RNA polymerases in general.
I saw a lot that it specifically works with RNA-dependent RNA polymerases, but I'm just wondering if it works with DNA.
It's possible, but the rate at which it does, or the effectiveness at which it does, is probably much less than what we find with the RNA-dependent RNA polymerases. I mean, that's true for AZT too, which was the first anti-HIV drug. It works primarily with RNA-dependent DNA polymerases. However, can it be accidentally used by the host DNA polymerase? Yes. Is that common? No. You have to have a couple of things occurring at the same time. One, those cells have to actually be proliferating. Two, the mistakes don't get caught by the repair machinery or the polymerase itself. That greatly reduces the likelihood. And if it just doesn't bind to the active site very well, that might be a third reason. So those would be my thoughts. I don't know, and this would be actually something that I'd have to look up. I don't know if DNA polymerases and RNA polymerases recognize something unique about the sugar, right? Because deoxyribose is different than ribose, so I don't know if there's a difference in recognition between DNA polymerase for deoxyriboses and RNA polymerase for ribose sugars. It's a really good question, though, but I have to imagine that that's been looked at. Okay. So here's what I want to do. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Okay. How many did I number off? Oh, perfect. Groups of 2. what I want to do is I want to have one group focus on the frequency of mutation, which isn't a figure but is an important part to the paper. Okay, so who would like to look at just the frequency of mutation of Ebola virus and GFP? Nobody. Okay. Sabrina, you get it. You and Morgan get it. All right. Figure 1. You have to describe for us the mutation frequency.
Like what you asked the question about.
Yes. And then explain the importance of it. All right. Figure 1. Am I just going to start choosing at random? It's going to be based on who looks at me. Cammie, you get figure 1. Figure 2. Michaela. And whoever you're partnered with. Figure 3. Andrew, whoever you're partnered with. Figure 4. Who doesn't have one yet?
Is it just figures?
Yep.
Okay.
I just want you to describe what's happening in the figure and explain and interpret. That's all we're going to do. Okay? Figure 4. Not for Grace. Grace, you get figure 5 then.
Thank you.
Okay. Who doesn't have a figure yet? Or a partner? Amelia. Why don't you take figure 4? It's a really hefty one, but I'm not expecting you to explain every part of it. Just summarize, okay? What?
We need a figure.
I didn't give you one? Then you get figure 6.
Oh, okay.
I don't know, whoever you picked. There is going to be one group of 3. I think it should be with Bill and Cammie. Well then,
Wait, so then do we need
nope,
5?
nope, we're good. We're going to make it work.
Then what should we do?
You guys are talking about figure 6. Cammie, what figure did I give you? 1? Oh, that's the easiest one in the paper too. Bree, you want to join them? Or is Amelia joining them?
I don't know.
I don't know either.
Well, but then no one's doing 4 if I join something, right?
No, you do 4. Do you not have a partner?
No.
Force Bill to do it. It doesn't matter. You're gonna do figure 4 with her. Cammie, you and Bree can do figure 1.
Okay.
4.
No, 6.
Okay, we'll do 6.
All right, we're good. Go. You got 5 minutes.
Oh, okay, so figure 5.
That 1 out of every 4.1 times 10 to the negative 4 nucleotides had a single nucleotide polymorphism, and that was over the GFP region, and then over the wild type region, the limit, it was below the limit of detection, so there's more mutations in the GFP region than actually in the wild type region.
Okay. So here's where I have a problem, because if you put 10 to the negative 4th, it implies that it's 1 over that number. And as Shania and Angela and Taylor pointed out, that actually means that the mutation rate, as reported in the GFP, would be lower. But apparently this is a product of how this stuff is reported. I completely disagree with it. The way that email said we should report it is like this: 4.1 times 1 nucleotide out of every 4.1 times 10 to the 4th, so 1 out of every 41,000, or 1 out of every, what is it, 91,000? 9.1 times 10 to the 4th. So we can say with this that the mutation rate of GFP relative to Ebola virus is at least 2-fold more. Right, this is our limit of detection, so it could be even lower than this, but they just can't tell given the technique that they used. How do they determine the limit of detection? Yeah, so they throw in an RNA that they know the sequence to ahead of time. They reverse transcribe it into cDNA, and then given however mutations occur there, they call that their background threshold, essentially. So how many mistakes would be generated by reverse transcriptase itself? Once they know that, then they've got their background. Okay. Grace.
One part that I was kind of confused on is, oh gosh, where did it go? It was like, oh, the range of, no, sorry, I gotta find it. Oh, the mean depth of coverage.
Yeah, so that stuff is, and so this kind of gets to the idea of what the heck is ultra deep sequencing.
Mm-hmm.
So deep sequencing or is a next gen sequencing. It's basically what we've done, but the ultra deep implies that you're getting like hundreds upon thousands of reads for any single nucleotide. That's what it refers to.
Okay.
So the coverage here, what they're saying, so for GFP, for example, they say total of 3.9 times 10 to the 5th nucleotides were sequenced. In other words, of all the GFP they looked at, they sequenced 390,000 nucleotides of GFP.
Yeah.
With a mean depth coverage of 670. In other words, each of those nucleotides was read an average of 670 times.
Oh, okay.
And the range of that was between 1,000 times and I think 330 times or something like that, roughly. Okay? So that's what they're meaning by that.
Okay.
And then they report somewhat similar statistics for the Ebola virus genome at the same time. Okay. So what's the interpretation here? Given these mutation rates, not only do we see... Let's take this a step further, I guess. The interpretation is mutation frequency is lower in Ebola virus than in GFP. What accounts for that? Why might that be? Go ahead.
So, they said that it's likely that most of the mutant genomes in that package or the genomes were not viable and therefore they were not harvested for the sequencing assay. So then they wouldn't be represented when they sequenced it. Therefore those mutations wouldn't account for the mutation frequency of the Ebola wild type.
Yeah, and in other words, there's a selective disadvantage to these mutations, and that's why they don't observe them as frequently. For GFP, there's no selective advantage or disadvantage with respect to the Ebola virus genome for this. They even say that they identified they think that there were multiple Ebola viruses that they harvested that actually lost the GFP altogether because there's no benefit to having it around. There's no selective pressure. But there is a selective pressure for the Ebola virus genome to be the way that it is. Right? So changes to the genome, which would alter changes to the amino acid sequence, are probably actually, instead of making Ebola virus more fit, are actually rendering it to be less fit. So take this back to the introduction real quick. In the introduction, they say that one of the reasons why this should be looked at is, where is it? Can't remember exactly where this is at, but they made an argument, I thought, that it's important to understand this. Oh, it's in the second paragraph. So it has been thought that Ebola virus did not evolve in context of a single outbreak, but recent data from West African outbreaks suggests that there is evolution. Filoviruses, which includes Ebola virus, have a wide host range, show great potential for emergence to new geographic locations, to new hosts, and to new modes of transmission. In other words, those new modes of transmission, new hosts, etc., evolution or mutations are going to drive all of those events. And so if those mutations can be accepted, I suppose, that makes the risk of Ebola spreading more likely. This would argue that maybe that the ability of Ebola to evolve to gain access to different hosts, to different geographic locations, is not all that likely because, at least in what they're observing in the virocells they're working with, these mutations aren't well received with respect to the virus. Okay? Sabrina?
Doesn't that seem kind of contradictory to what you would expect because it is an RNA virus?
Yeah, it does kind of seem contradictory, right? Now, I guess those mutations might be more tolerated if we were to force it, right? And we can force it in with certain artificial selection. So we can make Ebola go from a non-virulent form to a virulent form in guinea pigs by just passaging it in guinea pigs 3 to 4 times. And that then mutations have to accumulate there to allow it to adapt to that host. So you're right on that. And I don't know, maybe I'm over analyzing the data here. That's completely possible. But at least within a cell, within one particular host type, these mutations don't seem to be tolerated. That's what this result would suggest. Not like HIV, where I think those mutations are much more well tolerated.
That's what I was thinking.
Yeah. All right. Let's go on to figure 1 then.
So we talked about rising urine concentration increases the direction of the infectious virions decreases, but then we were reading about it more, and it talks about cytotoxicity not being observed. So is it because they're trying to get a replication interruption and not necessarily actually fully?
So the cytotoxicity refers to the effect on the model organism, or in this case, to the cells. So with any drug, if you apply enough of it, it's going to cause what we might call like off effects or side effects, right? You look at any commercial for any drug that's being sold, and they say side effects include, and if you're on antidepressant medication, one of the side effects always is suicide, which just seems so damn contradictory. But then there are other ones, right? Diarrhea, vomiting, nausea, all these fun things that usually get listed. Those are toxic side effects. That's what they're kind of referring to here with the cytotoxicity. So the amount that they're applying is inhibiting the replication of the virus, but not enough to cause death to the cells that they're doing this testing in. So that's what they're implying. So they identify how much is needed to inhibit half of the viral replication.
27 micromoles.
Yeah, and so that would be right about, I mean, it's somewhere around, oh crud. Nope, I want that. Somewhere around here. And note that this is a log 2 scale, so 1.5 logs is about 30 micromolar. Okay. Now, this S-shaped curve here is not unusual. You will see this with most drugs, where little doesn't have much effect. You need to reach a certain threshold to have the majority of the effect, and then much above that has no additional effect. I have a problem with this figure. Not the only one. But here's my problem with it. They don't even test concentrations below what the half maximal is. They've basically created a best fit curve and made an estimate based on that best fit curve. If I were doing this experiment, I would say you want to test at 3 micromolar, 10 micromolar, 30 micromolar, and then do the rest of these higher concentrations as well to show that there's actually an inhibitory effect. But all they have is 0 and 100 micromolar as their first 2 points. When we're creating standard curves, we want to make sure that whatever data we're extrapolating is within that standard curve. They're extrapolating a value here that's outside that standard curve. Okay? So that's the problem that I have with this figure. And that's how I would go back and do it. Okay. So what accounts for these effects then? What likely is contributing to this decrease in concentration of the Ebola virus? That one. This can go for anyone. Take a look at the paper. What do they suggest happens, Jonah?
Well, it could either be stalling the RNA polymerase, or it could just be mutating the genome to like a catastrophic
Yeah, and they give evidence towards that second one. So they actually take a look at the mutation frequency again in GFP and the Ebola virus genome when you're adding ribavirin. I can't remember what concentration they add. 500 micromolar. So it's way above. It's way out here. The concentration that's completely blocking viral replication. And what did they observe? Well, mutations are occurring at a rate of about 1 every 130 nucleotides by this point. Okay? So, absolutely, as you said, this is an error catastrophe, basically, that they're observing. Be nice to see if these events are also occurring at that ideal concentration, or that concentration that inhibits half of the viral replication. All right. We go to mouse models. What do we observe? And first off, I need to point out that this would probably be done in a BSL-3 lab. All right, so full protective equipment. The mice, even if they're living at the end of this, would probably still be anesthetized to prevent any potential spread of the virus. So I just wanted to point that out. Figure 2. And I've only included part of it. But what's going on? Okay, go ahead, Michaela.
We have, there's mice infected with Ebola, and then an hour later they were treated with ribavirin. As you can see, what was it?
It was ribavirin and then
Prior
yeah.
to it. Okay. And then you can see with all three graphs, the size up there, the 80 milligram per kilogram and the 50 milligram per kilogram, and the 0, they all exhibit a decrease in weight in the beginning. And then the mice, unfortunately, die a lot sooner without the treatment compared to the other two strengths. But it basically just reiterates the fact that ribavirin increases the survival rate and then prolongs death.
Yeah, so there's an increase in weight again, suggesting that, I mean, usually, what would weight be a surrogate measurement of in this case? Like, think about,
Dehydration.
could be dehydration, or if we're sick, vomiting. It could be a measurement. A weight loss could be a measurement of just not feeling well, right? I mean, I'm always excited when I vomit from getting stomach flu because I'm a little overweight right now. That sounds horrible.
you're
Sorry.
not.
No. I'm just going to stop this conversation right now because that really sounded bad. Anyway, I love Ben and Jerry's too much. Okay. So, but what we would find is a regaining of weight afterwards, right? So that's not atypical. The decrease in weight doesn't really differ between the two groups, right? At least over the period of time where we do see that decrease. And absolutely, as you point out, well, this curve doesn't continue because they're done. Grace.
So is the weight loss saying that they do feel some of the effects of Ebola? Or
I think that's exactly what it would argue.
okay.
Absolutely. So what that would suggest is that there is an active infection going on. But whereas those that are not treated with ribavirin are dying, these ones are basically becoming healthy again. I mean, that's the other way we could look at this, is that weight gain is a sign that they are becoming healthy. Yeah. What do we see in figure 3 then? So let's tie these two together.
So figure 3 compares 3 different treatment groups with the control group, and it shows in the control group rather, all of the mice die after 7 days, whereas treatment with both 50 and 80 milligrams of ribavirin result in different proportions of the mice surviving. And it said the difference in survival between the two groups were not statistically significant, but it appears that the proportion of mice in the 80 milligram group had less of a survival rate. And
Right.
We were thinking that could be because it's just too much of the drug to get in the mice.
Cytotoxic effects. Yeah, absolutely. So this is where we might see some of those off effects actually, instead of being helpful, probably exacerbating the problems that are already created by Ebola virus itself. So maybe a side effect is weight loss or something like that. That might prevent the organism from, in this case the mouse, from, I don't know, from basically returning to a healthy state afterwards. Go ahead, Andrew.
And another thing was for figure 2, like the weight loss, the error bars for that 80 mg group are huge. So
Yeah, absolutely. And that is a really good point as well. And so that would argue that maybe one of the toxic side effects are nausea or, not wanting to eat because of how the medication makes these animals feel. I don't know mice enough to know. I know they don't have a gag reflex, so they actually can't vomit.
Oh.
That's why you can basically take a tube and feed bacteria or viruses right into the stomach.
Probably poop it out then.
Yeah. Anyway. Excellent point. What kind of, so this is sometimes referred to as a Kaplan-Meier curve. If you look at clinical trials, that's what this would probably be referred to as. They do the statistical test here is it's the Mantle-Cox test. Another name for this one is a log-signed rank test. Basically what it means, or the way that this test is used, is typically in these types of trials where you're looking at survival between two groups. So you're looking at what point do they reach the end point, which in this case is death. Based on the trial, the end point could be cervical cancer. Right? If you're trying to determine does the new HPV vaccine actually prevent cervical cancer, you can look at two groups, ones that received the vaccine, ones that didn't, and make that endpoint cervical cancer. And look at percent that don't have cervical cancer over that period of time, compare the two groups. That's what that p-value there is representing. Okay? It's pretty easy to visually see it, but the statistics are there to support it. For all of you in 480 right now that love statistics. Bill. Alright. Bill and I are having fights not with each other over statistics right now. Anyway. Alright. What's that?
All sorts of fun.
All sorts of fun with statistics. Alright. Let's take a look at figures 4 and 5. And I'm not going to worry so much about you describing how the setup here is, because it's very similar to the last experiment, with the exception of what in both of these? How does the treatment differ?
When the drug was given.
Yeah, the drug's given here afterwards instead of prophylactically. Okay? So, who you have 4, right? 5. Who's got 4? All right, Amelia.
So, what did they say, 1 day post-infection? So first graph, like one of those ones are given infection, so it's just groups 6 through 7, and then group 9 obviously didn't get any of the ribovirin. So 6 through 7 have both of them. And we kind of attributed this to like the health and well-being of the mouse is what they're testing here, and then like what dosage is like the optimal amount to clear the infection, I guess, based on how healthy the mouse is.
So, does the concentration seem to be different, or does the concentration seem to have an effect on weight between those?
I would say not necessarily in the weight loss, but maybe in the weight gain back.
Okay. Yeah, I could see that.
But they all roughly lose weight from like down to about 80 somewhere, right?
Yeah.
80% weight loss up until like 10 days, and then they all go relatively back up, except for 50, which seems weird because I thought in the one above that was the one that went
So,
more dramatic increase.
so yes, but yes. I'll just leave it at that right now.
Yeah, versus before or
Mm-hmm. Yep,
Just
that's the
interesting
key
that it
point.
changed afterwards.
So then what happens with survival here?
Those that were treated 1 day post-infection were found to have a higher survival rate and a higher median in day of death.
Okay. So again, median day of death would be basically of all those that they tested. I can't remember if these are groups of, I think they're groups of 8 mice apiece. So it would basically be the midpoint between when mouse 4 and 5 died. Right? So longer group survival and longer and greater overall survival as well is how we would describe this. How does this compare to the prophylactic treatment? So take a look at, for example, take a look at figure 5B, which I have up here with 3B.
More death.
More death in which?
In figure 5.
Right. So what's that suggest? Yeah, prophylactic treatment. So if you were somebody, let's say that this comes online, it's already an FDA approved medication. It just hasn't necessarily been approved for Ebola virus infection. So we know that it's safe. Relatively at the concentrations that are used for HCV. You are an aid worker, and let's say that this is in Africa because that's the only location where we've seen Ebola virus outbreaks. Who do you give this to? Who do you give ribavirin to? That would be a good group.
People whose family members are infected.
Absolutely. I think those would be the best ones to start out with. So those who are in constant contact and those family members of individuals who are sick. Get it to them as soon as possible. Absolutely.
Are they doing that?
No. No, I don't think so, because I don't think this was being used as a treatment during that last outbreak. In fact, this paper came out March 2016, which means the research was probably being performed at the same time that that outbreak was occurring. So I don't think so. It's a good question. You should email him back. He's very nice. All right. No, don't tell him that. Don't tell him that. All right, let's go to figure 6. Now, Jonah, just give us the general overview. Don't worry about alkaline phosphatase and all those other ones necessarily, but let's just try to focus on the overview.
Okay, so basically the first one is the amount of infectious particles in the serum of non-human primates. And you can see that there's like the control group and then there's the treated group. Basically the ones with the treatment have less, or it takes longer for their forming units to accumulate.
Right. Let's just focus on those two. Let's just do A and B.
Okay.
And then for B, the treatment correlated with the time of death. So the untreated group had a higher rate and earlier death than the death of the treated group.
Good. The rest of these are all looking at signs of inflammation, signs of infection. I mean, that's how we can kind of group it. So like the thromboplastin and partial time thromboplastin are looking at blood coagulation, which is a typical association with Ebola virus. It's not in all cases, but the majority of cases we see that. This they describe in the methods. You wouldn't know this unless you looked at the methods, and it took me reading through this last night again actually to pick it out. This they described as a pilot study. Anybody know how many monkeys they actually tested? 4. Yeah, 4 total. So the untreated is a group of 1. The treated is a group of 3. Now, again, this data, how it's presented, I think can be misleading. All right? And that's just because the numbers probably don't support the use of these types of graphs. So this is one of those cases where when you're reading the literature, you want to make sure to know exactly what they're doing, so that it helps you to determine, like, do I actually believe this or not? Does the data look good? Yeah. Is it statistically significant? No, because they haven't had a large enough population. Maybe if the population of monkeys, non-human primates that they were using was larger, maybe they would have statistical results. They probably got money to do a pilot study for this before they just started infecting monkeys with Ebola virus to test it out. That's my guess. Okay.
Just out of curiosity, how much do you think a monkey costs for research?
I don't know. Why don't you call up Covance? Because that's where they got them from.
Alright, what's the number?
I don't know. I don't know. I honestly don't know what the going rate for a monkey is for research.
I think Brenda said it was like $1 million per month.
$1 million per monkey?
Yeah.
Holy mother of pearl, that's more than a dog. Okay.
More than a dog.
I'm going to shut my mouth. See, there was a reason why I wasn't talking today. All right. So, I just want to point out real quick then, I think this is the last point that I wanted to come to, and just to kind of close this up, how might ribavirin work against Ebola virus? And this takes us back to the original idea. Probably by mutation, maybe by blocking replication, though we can't necessarily tease those two apart in this particular experiment. Okay? One thing to kind of think about more broadly are, what are the other targets? I mean, this is a target where you're actually kind of indirectly targeting the RNA polymerase. But what would be other potential targets for Ebola virus? And I'll kind of leave this one open right now for you to just think about. But think about what actually causes the signs and symptoms of Ebola. Or what might we want to do to enhance the immune response would be another way to think about this. And that kind of goes along with the next one. What characteristics might you want? Okay, so part of the reason why I've forced you guys to do this way, I think, I'm not committed to this yet, but I think this is going to be the last lit review that we have for the semester. Okay? But, oh, don't look so sad, Sabrina. I appreciate that, but, there's always a but. Your final take-home is going to be to read a paper and to write a review of that paper. I've not come up exactly yet with what this is going to look like. I'm going to give you options of papers so that you can choose what interests you. I'm going to probably pick 3 or 4. You decide the one that best suits you. But it's my way to see, what the hell have you learned from all of this that we've done throughout the semester? I think it's a really good formative assessment of what we've done throughout the semester. Okay? So look forward to that. Of course, like every take-home, you'll have at least a week to work on it. But I just wanted to give you a heads up. All right, that's it. I will see you all on Friday, BYOM. Sabrina, don't get sick.
5,000 and 15,000.